Hi everyone, hope you are well. My name is Akshay Agarwal and I will be presenting our work on discovering proteins, function to name. All of this work was funded and performed at IBM Research and wouldn't have been possible without all the co-authors. So the overall problem that we are trying to tackle is naming or classification of hypothetical protein sequences. For this, we are first trying to predict their function and then use that to identify which protein it's similar to or if it's a new protein altogether. I'll start by presenting the problem and the background. I will then move on to discussing how we predict function of protein sequences. Then we'll discuss how we can perform biological validation and opportunity for a deeper dive. And lastly, I'll summarize my results. So hypothetical proteins are regions that are identified as potential proteins by annotation tools such as PROC or MAF. However, their name and often other characteristics, such as function, is unknown. So for example, we might not know which pathways that hypothetical protein belongs to. We may not be able to predict its structure, etc. So in our group, we rely on PROCA for protein annotation, that is annotating a genome sequence to list its gene or protein sequences. And for functional annotation, we use InterproScan. To give a quick introduction, InterproScan takes as input an amino acid sequence and references 15 plus databases to annotate the functional domains present within that protein. InterproScan is able to annotate some hypothetical protein sequences, however, there's still a significant number left. Functional domains are catalytically active subcomponent of a protein and indicate bioactivity of the organism. This describes the phenotype rather than the genotype alone. For our work, we construct a domain architecture for each protein sequence. Here we sort the domains, check whether they are present in a protein sequence or not, and construct a vector indicating the same. This also helps us reduce the dimensionality since we go from a DNA sequence to amino acid and then to domain architecture. So after having prepped the data and annotated the protein sequences and after constructing the domain architectures for each protein that InterproScan was successfully able to annotate, we get to the part where we start building and testing our system for classification of hypothetical proteins by function. So for classification by function, we have two major parts. The first is building a classifier for predicting IPR codes, and second is performing biological validation. This is not to be confused with machine learning validation, as here we have more interesting things in store for you. So here we talk about the motivations of performing prediction. As I said previously, Interpros can annotate some hypothetical protein sequences, however, not all of them. This creates a problem since we need function to classify our protein. Thus, we decided to construct our own functional predictor. In order to construct this functional predictor, we leveraged 27,000 IPR codes, each with at least one domain sequence. We use 70 million gene sequences which are present in the functional genomics platform. This is maintained by IBM, specifically by my team. These 70 million gene sequences contain approximately 40 million named DNA sequences which are used for training and testing, and about 30 million hypothetical nucleotide sequences which are to be classified. Now, I told you that we want to annotate our hypothetical proteins by finding out their function first. However, in our initial approach, we had built an ML model to directly assign name. I want to share some things that we learned from it. Now, even though the model had a very good area under the curve of 0.91, it was only able to classify in the context of the named proteins that it was trained against. And also, it wouldn't be able to discover any new proteins. Second, it was very difficult to perform biological validation of the results since the model was essentially a black box. Other than this too, it was plagued with some other problems such as training on a dataset with unequal class sizes, then the requirement to be trained differently for every genera, etc. So we started by developing a system that can predict IPR codes for a sequence. Given the shortcomings of the machine learning approach, we decided to modify our method in two ways. First, instead of predicting name directly, we tried to predict function, more specifically IPR codes or domain architecture. Secondly, we use Kraken-2, a taxonomical classification system using exact gamer matches to achieve high accuracy and also fast classification speeds. In Kraken, we need to create a taxonomy. So the first flow in our process is making this taxonomy. This taxonomy is used as a database by Kraken in order to perform predictions. Without going into too many details, I would just say that the lowest common ancestor technique is used to create this tree. UID corresponds to a domain that is found in one or more protein sequences, and IPR code is the interproscan functional code. For domains that are shared by multiple IPR codes, we LCA up those k-mers and combine the IPR codes. In addition to this taxonomy, we also provide Kraken with the domain sequence for each UID. I would like to highlight that we constructed this taxonomy using 250 million domain sequences, resulting in a tree which had more than 15,000 nodes. So once Kraken has all the information that it needs and is trained, we treat this as a multi-label classification problem in ML to perform evaluation. It thus evaluates its performance by treating each IPR code as a separate tool and calculating metrics such as micro-precision, micro-recall, and F1 scores. Mainly, we depend on ROC curves and area under the curve to analyze the performance. We see that in this particular case, the area under the curve is 0.87. Furthermore, from the ROC curve, we are able to identify the threshold by using the needle algorithm. Once we have the threshold identified, we put all of this to work. So we get a nucleotide sequence that we pass to Kraken2, which breaks it up into k-mers, performs a translated search, and reference taxonomy tree to give a list of IPR codes with a score. We then threshold each score and get the final set of IPR codes. Right now, the prediction is done using Kraken2, but this component can be developed in a different panel by leveraging more advanced machine learning techniques. We've talked about how we've performed the prediction process. Now I would like to share some of our early results using domain architectures to better characterize hypothetical proteins. This is prior to the Kraken prediction process, and we resolve hypothetical proteins that have already been successfully annotated by Interproscan. We've reviewed how this domain architecture is constructed, and we observed that out of the total 25 million unique protein sequences that are named as hypothetical protein, approximately 14 million proteins already have domain architecture that can be used to further characterize their function. These 14 million protein sequences distill down to 48,000 unique domain architectures. We can then map by complete set identity to other named proteins by matching these domain architectures. So finally, we see that approximately 64% of the proteins map to a predominant protein name, where 50% of the total set matches to exactly one protein name. The remaining approximately 36% have a mixed distribution of protein names and provide a more complex challenge to resolve. Now, this is only for the 14 million proteins out of the 25 million total that had some interproscan annotations, and for the 11 million, we would need to use the Kraken prediction to get the domain architecture first. Now, next we come to performing biological validation. As I said, this is not to be confused with machine learning validation. The idea here is that beyond the evaluation metrics, how can we perhaps get a little more intuition about the functional space in general, and then about the predictions that we are making. So since our space has a huge number of dimensions, where I said that it's about 27,000 IPR codes, it's very difficult to visualize it. So the first thing that we did was run PCA on our dataset so that we can bring down the number of dimensions. After running linear PCA, we obtained 4 principal components, which explain more than 80% of the variance. This is our first iteration. I think we need to try this with different kernels and also look at dimensionality reduced results more closely to identify if TSNE might be a better choice or if we want to do something entirely different. So once we have the dimensionality reduced, we represented the 30 million proteins in the functional space. Most of these points are named proteins for which we have obtained functional codes from Interproscan, and some are hypothetical or putative proteins which are also annotated by Interproscan. Right now, we are not quite at the stage where we put the points classified from Kraken, since we are still working on refining the results further and on assessing our functional space. This space helps highlight the difference between phenotype and genotype. If viewing by name, it shows that name alone does not describe their function, i.e. named proteins aren't clustering. The function is what is driving the clustering. But that's not all it can help with. I'm going to first show you a short video of how big this space is for now, and then I'm going to talk more about a few ideas regarding how this can be used for analysis. One thing I would like to say is that this space is a representation of the technique of the statistical data. So most things can be done by using statistical methods. However, this visualization helps form more concrete hypotheses and get a better sense of what's happening. So in this video, we want to reiterate that the three dimensions or three principal components are represented by the three dimensions, and the fourth dimension is color. I'd like to point out that we observe an increased density of points along the y-axis, and as we zoom out, you can see how large the space is. In fact, as we keep zooming out, we see that a lot of clusters are at the periphery of this space. We believe that it would be worthwhile to start understanding by evaluating these points, since they have a greater degree of separation from the other points in space. This helps provide visual context on phenotype versus genotype, and by looking at these clusters, we can also validate our visualization. Example, if clusters for same protein name are vastly separated, then does that say something about the protein or about us messing up our analysis and dimensionality reduction? So this is a great example of what we showed in hypothetical protein characterization, where the surrounding points are uniform in green, and it's easy to classify the hypothetical protein, which is indicated by the purple dot. However, in this particular case, we see that on the left there are three shades of blue that have one protein name but with varying domain architectures. The yellow dot indicates the hypothetical protein. In this case, since we do not have an exact overlap between domain architectures, it warrants investigation into what this hypothetical protein tells us. For example, is this protein evolving differing function due to a favorable environment or perhaps stressors? What should be the appropriate distance threshold for this hypothetical protein to have the same name, etc.? On the right, we observe a similar trend where the domain architectures are even more separated. So the final take-home points are that we present multiple methods to address the annotation gap of hypothetical proteins. One, you can use prediction using functional taxonomy and sequence-based analysis. Second, you can do protein-to-protein comparisons using domain architectures. And third, you can do large-scale visualization of a complex protein space. Together, these methods aim to better characterize hypothetical proteins in the face of missing data. Thank you so much.